I'm here today with Dr. David Cooper from Massachusetts General Hospital, who is a world-renowned expert in transplantation. And today we're going to speak about his new paper, Immune Response and Inflammation in Allo and Xenotransplantation. So welcome, Dr. Cooper. Thank you for taking the time to speak with me today.
My pleasure.
So first, can you tell me a little bit about you and who you are and kind of your background in transplantation?
Yes, sure. I was born in London, England, and I had all my medical student training in England. And I decided to go into cardiac surgery, which was just beginning at that time. It was open heart surgery was fairly new, and I was influenced by two very good surgeons at the hospital I was at in London, Guy's Hospital. And then I was very interested in research, and so I started a PhD. I did a PhD looking at heart transplantation in the very early days of transplantation. Actually, before any heart transplants have been carried out in patients. And when I finished my cardiac surgical training, I wanted to stay in an academic position with some research, and I couldn't find one in Britain at the time. There were not that many there. And I wrote to Chris Barnard, who did the first heart transplant in South Africa in 1967. And I wrote to him and said, can I come and do some research with you for a year? And that led to me actually staying there for 7 years, partly because his brother, who was one of the surgeons, retired early, and he offered me his brother's position. And so I stayed there. And then through him, I moved to Oklahoma City in the States, and I continued my research into xenotransplantation, the cross-species transplantation. And since then, I've been at Mass General and the University of Pittsburgh, University of Alabama, and back now at Mass General. So I've moved around quite a lot.
Wow, what an exceptional career. And it's so cool. So you just reached out to him and were like, I want to get involved, and he welcomed you to do that. That's amazing.
Yes. And in fact, he had a small research group there with particularly a biochemist, and we did some very good work there on management of brain dead donors, and found out quite a bit then, and introduced thyroid hormone to maintain their stability, hemodynamic and metabolic stability. So that was a very exciting time too. And running the transplant program for him, as I did in Cape Town, it was still very much this was 1980, it was still very much a research program. And so that fitted in with my wish to be at the sort of cutting edge of things. And so I think that was a sort of clinical research. But at the same time, we had a very good research laboratory, and I was able to do some interesting studies in the lab.
No, that's fascinating. It resonates with my personal experience. I reached out to people early on in my career and was just amazed at the reception that happens. And so I think it's really important for us to let everybody that's a new investigator and young investigators in transplantation know, hey, nobody's so scary. They're not going to kind of like
No.
want to help you out. And I think that's a really that bold move that you made certainly did set your career apart. And I think that's important to remember for the young
Well,
people.
many of the research fellows I've had attached to me in my career have been people who wrote to me and said, can you take me for a year or two? Or their boss has written to me or something like that. So I think it's very common these days to do that.
That's fantastic. So let's talk a little bit about this paper that you have just published in Transplantation. And I'm not a basic scientist, so some of this is kind of a little intuitive to me because I hear these words inflammation, and in the media today we hear inflammation is bad and you want to reduce inflammation. So can you talk to me a little bit about what is inflammation and why does it matter in the context of transplantation?
Well, let me first say that the initial draft of the paper was written by Mohammed Ubaid, who was new to this country, and while he was actually doing a clinical position elsewhere, so he did a very good job of it. And then the other co-author was Mohammed Ezelarab, who was a former colleague of mine in Pittsburgh, who actually is an authority on inflammation in xenotransplantation. So inflammation is a very non-specific thing. It can happen in all sorts of conditions. I mean, if you have trauma, any sort of trauma, you get an inflammatory response. I presume it's a protective response, evolutionary. And if you have an infection, you get an inflammatory response. If you have a transplant, you get an inflammatory response. And let's say you rub your leg against the wall and it goes red afterwards, that's an inflammatory response. So it's a response to all sorts of incidents that can affect our health in one way or the other. There are diseases that are thought to be inflammatory diseases like rheumatoid arthritis have sort of got an inflammatory background, and so on and so on. And it was well known that you had an inflammatory response for any surgery, for example, like a kidney transplant. And my group, Mohammed Ezelarab particularly, we identified a very major inflammatory response to the presence of a xenograft, a pig organ graft. Presumably that is related to the fact that the body is not used to having a pig organ inside it, and it responds to it as if it's an invasive thing. And that can be quite prolonged. It can go on for several weeks. And we think it's important, particularly because inflammation can augment your immune rejection response. So it increases your rejection response. So we now treat the inflammation as much as we treat the rejection. There are certain drugs that will suppress certain cytokines and other agents that are prominent in inflammation. And we try to suppress those as well as give immunosuppressive drugs that suppress the immune rejection response. So we have to deal with it in several different approaches.
Okay, I think I understand. And so how is this inflammation different between allo, right, human organs, and xeno, non-human organs?
Well, there's a lot of overlap between the two, but we were impressed by, when we thought about it, of the causes of inflammation in the two. For example, in an allograft, particularly if you're from a brain dead or circulation cardiac death donor, that brain death is an inflammatory elicits an inflammatory response. So you're taking an organ out of somebody, that the brain dead patient or the circulatory dead patient, who's already got an inflammatory response, and that can affect the organ that you're taking out. You're putting it into a patient who may have been on dialysis for several years and have a number of comorbidities, say some heart disease or something. They all can give you dialysis, and comorbidities can give you an inflammatory response. That's one reason why a lot of people take a drug like one of the statin drugs, because not only does it lower your cholesterol, it's also anti-inflammatory, and that's thought to be good. And then you have the transplant, which is an inflammatory. There's an inflammation to the surgery. So you have a bunch of these things causing inflammation in the patient after they get an organ transplant. Now, in xenotransplantation, you can have far fewer of those, because you will take out the pig organ from not a brain dead pig, but just a healthy anesthetized pig. It'll be a younger pig. It won't have had problems itself with its health. It won't have an infection that might be inflammatory, and so on and so on. Whereas many human organs have come from a patient who's had an inflammatory response even before they were brain dead. And you're putting it into a patient who needs a kidney transplant, but you'll be able to put it in much earlier in the course of the treatment. You could actually put it in before they ever go onto dialysis. So all the factors that may make inflammation in the patient, dialysis, length of time they've been on dialysis, their comorbidities, maybe they will be minimized. So there should theoretically be a much less inflammatory response to a xenograft than to an allograft. However, we did document that there is quite a major inflammatory response to a xenograft, irrespective of anything else. And I think that's because it's a foreign tissue and the body recognizes it differently. So you have a difference in the causation, but there's a lot of overlap in what it causes, what the inflammatory response it consists of.
That makes sense. And I guess that really makes a difference for long-term graft survival, regardless of if it's a allograft or a xenograft, right?
I presume it is a factor. I'm not sure that anybody's actually showed that conclusively, but certainly inflammation, as I mentioned earlier, can augment your immune response, your rejection response. And so if you have a lot of inflammation, you're going to get other problems. It activates your immune system, so to speak.
Okay. So can you explain to me what inflammatory biomarkers are?
Yes, they are. There's a whole host of them, and they're just something you can measure in the patient's blood that indicates they are undergoing an inflammatory response. One of the commonest ones is called C-reactive protein. It's produced by the liver, and this is present whenever you have an inflammatory response from whatever the cause, really. And we see it particularly in non-human primates with a xenograft. And if you give an anti-inflammatory drug, you may suppress that C-reactive protein, so the level goes down. So you know that you've suppressed some of the inflammation. But there are a whole host of others, some of which we use and some of which we don't use much. Histones, cytokines, all sorts of things you can measure in the patient that will tell you whether there's an inflammatory response. So we call that a biomarker. And if you select a bunch of biomarkers, you get a good idea. Are you suppressing the inflammatory response, or is it still raging ahead? And clearly, we'd like to suppress it if we can.
And do we do those tests on your average transplant patient regularly?
I don't think they do. I haven't been in clinical transplantation for a number of years, and I was never in kidney transplantation. I was a cardiac surgeon. I don't think, I certainly didn't do that in the old days. We do it pretty regularly with the xenotransplants, certainly in the animal models. I'm not sure how much they do in the patients, the few patients that have been done, but I think it would be a good idea to follow the inflammatory response. Are you still suffering from an inflammatory response, which is causing perhaps more rejection, so on, or have you got it suppressed? We do routinely, though, now give drugs which suppress the response. Doesn't necessarily suppress the whole of the response, but it's that these drugs have a major effect in reducing the response. So we try to control the inflammatory response as best we can.
Okay, so given some of these unique barriers that we've already seen in xenotransplantation, how do you see current immunosuppressant regimens shifting or adapting, or are there things that we're still learning to kind of be a strategy for these patients undergoing xenotransplantation?
Well, the important point is, and we observed this, a research fellow of mine, Leo Bueller, in the year 2000, long time ago, we noticed that the drugs that we commonly give to patients with an allotransplant are not very effective at reducing rejection or preventing rejection of a xenograft. But a new form of drugs, which are just beginning to get into the clinic now, it's taken 20 or more years to get that far, are very much more effective. And I think they're wonderful drugs because they seem to me to have very few side effects. And we've used them in the monkeys and the baboons and so on, and they've used them here at MGH in the first 3 kidney transplant patients. They're a drug that doesn't block the actual T cell response, which is one of the key cells that causes rejection, but it blocks the stimulation of the T cells. They're called co-stimulation blockade, and they're very effective at preventing rejection. So in the animal models and in these patients, we haven't seen much rejection at all to a pig kidney. in recent months or even years. So we've come a long way, and I think it's primarily due to these new drugs. They're under clinical trials now in patients with allografts. One of the companies will have some results by October in the effect of it in kidney transplants in patients with allografts, and I say they are being used in the patients with xenografts as well. I think they're a huge breakthrough. For example, one of my colleagues, Robin Pearson, here studied them in a monkey heart transplant model. And if you give that drug with nothing else, once a week, you never get rejection, basically, which is quite remarkable. Just to give one, and you give it intravenously. That's perhaps one of the disadvantages in a way, that you have to have it intravenously. But for studies like xenotransplantation, it's probably an advantage because you're sure the patient's going to get the drug and not relying on the patient to remember to take the tablets and so on. So these drugs, I think, are going to revolutionize transplantation. They're also useful in other, or they're being tested in other conditions, such as rheumatoid arthritis and some other inflammatory sort of conditions or immune-related conditions. And I think they'll have a role there too. But they're quite remarkable, and I'm very, very impressed with them. And they have very few side effects as far as we can see. So, so we actually should move to these new drugs for xenotransplantation. There's lots of evidence they're much more effective in a xenotransplant than if you use the standard conventional therapy.
Wow, that's impressive. That's really exciting for the field.
Yes, I think it's making a big difference.
But you mentioned that they have to be administered through IV. So do they need to be done in the hospital? Is this something that
I don't think so. I think you could arrange for them to be done at home and so on. I don't know what these companies think. I presume eventually they're going to try to develop one that you could take orally or at least intramuscularly. But at the minute, we're using them intravenously. And I say they have they're dramatically improvement over the previous therapy we had, or the conventional therapy.
Well, that seems like it's a lot of hope for you all as scientists, and also for patients who might be trying to understand what's next on the horizon for immunosuppression. So that's really, really, really cool. So outside of that, and based on your review of inflammation, what key immune mechanisms still need to be addressed before xenotransplantation can match or kind of exceed the outcomes that we see in allotransplantation, particularly with regards to, rejection, tolerance, induction, things like that?
Yeah. Well, it's a bit early, of course, to know how long patients are going to survive with a pig kidney or a pig heart, because we haven't obviously followed them up. We have two patients here at MGH. One is now more than 6 months after the transplant, so that's encouraging. And the other one is a couple of months after the transplant, and they're both doing pretty well. One thing we've observed in the monkey model, the baboons, with pig kidney transplants, is that they don't now, as I mentioned, they don't now get much rejection. We seem to have been able to control that. There are no features on the microscopy and so on of the tissue, but at least half of them are getting a loss of protein in the urine, which we call proteinuria, and it can be quite excessive. And that does occur in a small number of patients with allotransplants, and it doesn't bode well for the future, because if you're getting proteinuria, usually your graft doesn't survive as long as if you're not getting proteinuria. We don't know the reasons why you get proteinuria. It's probably an immune response, not sufficient to cause rejection, but sufficient to cause some irritation to the kidney, so that you do lose protein, which you obviously should not do. And I think that's going to be quite a problem we have to sort out. We have to investigate it. Now, one good thing is that because half the baboons with a pig kidney get proteinuria, we now have a good model, experimental model, to say, can we sort this out? Because if we could sort it out in that model, probably it would affect the patients who get proteinuria after an allograft, or patients who get proteinuria from some kidney disease, which is not very poorly understood at the minute. And there are patients that do get a severe proteinuria. So I think we have this model that we could use to try to sort out what's going on. But I think the fact that we're getting proteinuria is going to set us back a bit. We have to overcome this problem if we want really good long-term survival. The other important point about that is that we've recently shown that if you're losing protein in the urine, you also lose this key novel immunosuppressive drug, which is a protein. That's also could be lost in the urine. And if you're not measuring the levels, the level will go down till you've not got a therapeutic level in the blood, and then you'll get rejection. And we've seen that certainly in one baboon, but after that, of course, we were measuring the level. But sometimes we would have to increase the dose very significantly to keep the level up. Now, it doesn't occur in everybody. It doesn't occur in all the baboons, it doesn't occur in all the patients, but it is an added problem. So that's something we've got to sort out.
Wow. Well, it seems like there's a lot of progress, and it really does seem like it's important that the transplant community care a lot about inflammation as a big concept, because it can lead to improved graft survival if we can solve some of these problems. And most importantly, and I think from where we're all really excited is, well, can Xeno actually live up to the promise that we've made it to be, right? Is it going to be able to be either a bridge or a long-term solution for the kidney shortage? And you're helping solve some problems that will ultimately, hopefully, allow that to happen. I think that's fantastic.
I think I'm very optimistic about the future, because although we still have some problems,
Yeah.
I think we're very likely to get better and better pigs for start, which have some protection against inflammation. We do have pigs that have protection against inflammation, but it's not total. And we have pigs which are genetically engineered to protect themselves or their organs from the immune response as well as the inflammatory response. I think eventually we'll have pigs that cover everything. We won't need to give any immunosuppressive drugs. Because the pig will probably express so many factors that will be protective. So I'm very optimistic that over the next 20 or 30 years, all these problems will become resolved. And the logistics of having a xenograft rather than allograft, surgeons don't have to get up in the middle of the night and rush out somewhere to take out the organs. You don't have to worry about have you got an operating room ready, because you can just plan this ahead. You can phone up the pig people and say, on Monday morning, I'd like a heart or a kidney at 9 o'clock. And the logistics will be so much easier. And patients will not have to be on dialysis for 5 or 10 years, which is $100,000 a year. They'll be able to transplant much sooner. So not only will it be easy for the patient, but the logistics of the whole thing, I think, will make it very popular with the hospitals and the surgical staff and so on as well.
Yeah, that's a great point. I mean, I think about it in the United States, and I have a kind of unique perspective, and I understand some of the rationale for keeping operating rooms, available at night, and revenue streams. And thinking about xenotransplantation and the opportunity to expand transplant access around the world, particularly to lower and middle income countries, with sort of like call up the pig company and they're going to send me my kidney or my heart for my transplant patient. Do you see, a lot of are you inspired by that concept? Do you find that we need to do special work to make that happen? Where would you
Yes.
say we need to go next?
Well, of course, I think unfortunately it's going to be an expensive form of treatment anyway, just as allotransplantation is a very expensive form of treatment. And therefore, some of the third world countries will not be able to afford it. But if we want to make it available around the world, which I think we do, then we have to, one more factor we have to do is can we store these pig hearts for, say, 2 or 3 days without them losing any function, so that we can ship them from somewhere in the United States to Europe or to Australia or India or wherever we want to do it. So I think that's something that people are beginning to look at now. Can we store the organs better than the systems we have now? And if we could store them for, say, 3 days, I think you could send them anywhere in the world. And if when they get there, they haven't lost any of their function, which is obviously important, then you can supply them. Because when we have these pigs in these facilities, which are they're already in them for use in the clinic, those pigs are housed under very clean conditions. It's quite a detailed. They're monitored every now and then to make sure they don't carry viruses and so on that you don't want them to carry, and you won't be able to have those facilities too many of them because they're going to be expensive. So you need to have them placed in a situation where you can ship the organs to within a fairly wide distance. Initially, it may be a couple of thousand miles, but eventually halfway around the world if you want to. So I think that's something that we're obviously going to need to work on as well.
Yeah, I'm sure there's a lot of regulations and laws and all sorts of things that will come into place with different countries and how they manage things, even with air travel. So I think that's going to be an exciting frontier for the global transplant community to figure out. But first, we have to figure out inflammation and making sure that we have kidneys that work in people, whether or not they're from animals or from other humans. And your work really has been extraordinary in moving that field forward. I wanted to ask you one final question, and that is kind of outside of transplantation. What does this mean for the global picture of healthcare? Are there other conditions or other things outside of kidney transplantation that this work in understanding inflammation can assist with?
Well, of course, it will be valuable for any organ transplant. Heart transplant is a big inflammatory response to a heart transplant, partly because you have to go on a cardiopulmonary bypass, which is an inflammatory procedure. But it will be valuable for liver, lung, and so on. And particularly, I think one of the biggest areas where it will make a huge impact is in patients with diabetes, because you can transplant pig islets that make insulin. We did the first successful one back in 2009 in a monkey. Went for a year without any insulin. The diabetic monkey went for a year, and it's becoming increasingly possible now. And bear in mind, there are 30 million people in this country alone with diabetes of one sort or another, and well over 1 million of those have the juvenile diabetes, which is sort of life-threatening at times. So I think that will be a big, and there are other areas too. For example, some colleagues of mine in Europe some years ago showed that they could cure Parkinson's disease in monkeys by putting pig cells into the brains of the monkeys. And they stopped doing that mainly because the EU frowned on doing work in non-human primates. But that's another area where it could be. And then the third thing, which I think is we haven't really looked at yet, but I'm very keen on, is that we spend a huge amount of money each year on providing products for patients. For example, a lot of patients need serum albumin, and so they get human albumin transfused. Now that albumin is collected from blood from thousands of patients around the world. They bleed thousands of patients and isolate the albumin to give to patients. They bleed thousands of volunteers and isolate the albumin and then give it to them. I'm told that the cost of that per year is something like $30 billion. Now, I think we could do all that in a genetically engineered pig. We could make a pig that is genetically engineered to make human albumin instead of pig albumin. And then we bleed the pig once a week or once a month or something and isolate the albumin from the blood. And then we have a source of it. We don't have to go around the world bleeding thousands of patients. And every time, of course, you bleed a patient, you don't know what infections they've got that you might be bleeding. Whereas with pigs, you can monitor them. You know they don't have any serious infections. So I think there's going to be a huge need for genetically engineered pigs for these purposes, as well as for transplantation and treatment of patients perhaps with Parkinson's disease and so on.
Wow, that is just fascinating to me. And so a lot of your work has been funded by the National Institutes of Health, including this paper, as well as a Kidney X Prize. and some support from the American Society of Nephrology. So all these wonderful funding organizations putting resources towards work. This is what it's doing, right? We're learning from it. We're going to be able to change the world and change the way healthcare is delivered to patients. So I just wanted to say thank you, Dr. Cooper, for your pioneering work in this space. I know how important basic and translational science is to making transplants happen. And sometimes people like me and patients don't understand all those details, but you've really broken it down in a way that I think is really understandable. And so thank you for your time and for all the work you've done.
It's my pleasure. And I'd add, sometimes I don't understand all these things either. So you're not alone.
Good, good.
Okay. Thank you
Thank
very
you so
much.
much. Take
Okay.
care. Okay,
Bye-bye.
bye-bye.